Hello, and thank you for joining us for this Nantucket Tick Talk. My name is Yvonne Ballencourt. I'm the director of the Nantucket Field Station. We are a facility of the University of Massachusetts, Boston, managed by the School for the Environment. We sit on over 100 acres of conservation land that the Nantucket Conservation Foundation owns. We run classes. We also support a wide range of universities, agencies, and institutes that need to do research on Nantucket. You don't have to be part of the UMass system to be able to have access to our facility. We're about to hear from someone who has been using the Nantucket Field Station for decades. So without any further ado, it is my distinct pleasure to introduce Sam Telford. Take it away, Sam.
All right. So this is a talk that I gave first week of December last year. And for those of you who attended that, I apologize. There'll be a few new slides or emphases on those slides in this. But essentially what I want to do is highlight the history of tick-borne disease research on Nantucket Island and the central role that the field station has played in allowing us to accumulate knowledge about tick-borne disease. And in so doing, if we have that knowledge, we can try to use it to come up with ways of reducing risk of people getting it. I'm not going to bore you with the clinical aspects of the tick-borne infections. But you should know. If you do not know, you need to go to the Department of Public Health website or the Martha's Vineyard Board of Health website, where they have very good information on the clinical aspects as well as how to prevent these infections. But you should know that there are 5 different infections transmitted by deer ticks on Nantucket: Lyme disease, babesiosis, granulocytic anaplasmosis, Borrelia miyamotoi disease, and deer tick virus encephalitis. Actually, we have not seen a case of deer tick virus encephalitis on Nantucket yet. I'll come to that in a little bit. And I also put down a relative index of how important these are. Epidemiologists like to look at numbers per 100,000 per year. And so in a community like Nantucket, you can have 200 to 500 cases per 100,000 per year. Babesiosis, you have maybe half that, and anaplasma, maybe half of that, and then the other 2 are extremely rare. And so this varies from year to year, but in general, that's what we call the incidence of these infections. And to put it into perspective, STDs in the inner city can reach 200 to 500 cases per 100,000 per year. So it can be a burden to your community. Most people don't realize that before Lyme disease, there was something called Nantucket Fever. And that is human babesiosis, and it was exclusively found on Nantucket for the first several years. Nantucket, as a result, became a hotbed of research for tick-borne infections, and the ecology of babesiosis and Lyme disease was worked out there on the field station. Since then, we've prospected for new pathogens, this granulocytic anaplasmosis, Borrelia miyamotoi disease, and deer tick virus encephalitis. The field station has been part of our search for new things transmitted by the same ticks, and also has served as the foundation for some of the interventions that we'll talk about, such as deer reduction or the Lyme disease vaccine, and then a new one on genetically modified mice. So we've published over the years, and this all started at Harvard, at the Harvard School of Public Health in the mid-1970s. I came out of that lab in the late 1980s, early 1990s, and cumulatively, we published more than 50 peer-reviewed papers from 4 researchers working at the field station, working with Andy Spielman, the late Andy Spielman, who was professor of tropical public health at Harvard, Joe Peisman went on to be the chief of the Lyme disease vector section at the CDC. Mark Wilson is ecologist at the University of Michigan. And then my wife, Heidi Gertet, did her doctoral work on the field station with cottontail rabbit transmitted infections. And a number of major findings came out of our work over these, I hate to say it, 40 years or so on Nantucket, identifying the white-footed mouse as the reservoir for Babesia microti, identifying a tick as something new to the landscape, and we still consider it to be a different species. How does the tick keep itself going in nature, the life cycle of the tick over time? And then, believe it or not, the field station served as my home base in running the phase 2 clinical trials of the SmithKline Lyme disease vaccine, Lymerix. We also, working with Tim Lepre at the Cottage Hospital, identified the first case of granulocytic anaplasmosis and was able to quickly figure out its life cycle and how it coexists with Lyme and Babesia. And so these are not trivial findings. These serve as a basis for a lot of the understanding of the epidemiology of deer tick transmitted infections in the northeastern U.S. So a half century ago, actually it was a half century last year, it was July of 1969, when a 59-year-old woman came down with a malaria-like illness. She was a summer resident and went back, was convinced to go to a hospital in New Jersey where she was diagnosed with malaria, and the CDC was tracking malaria cases. And looked at the blood smears that were sent from this patient, and in a feat of pretty good microscopy, identified it as a rodent infection, Babesia microti. And it led to the case report in the New England Journal of Medicine, Babesiosis in Massachusetts Resident, which was published 50 years ago on October 15th of this year. Everything started then. And strangely enough, in the next 6 years, another 6 cases were identified on Nantucket. And then by the end of the 70s, people recognized that this infection occurred on Long Island, Rhode Island, Block Island, Martha's Vineyard, Cape Cod. But oddly enough, the Merck manual, in the old days before the internet, you went to visit a doctor, and you'd be sitting there, and he or she would say, oh, well, excuse me a minute. And they'd go back to their office and consult the Merck manual, which was sort of like Google for medicine in those days. And the Merck manual in the 70s specified a travel history to Nantucket Island when one was considering Nantucket fever. So it was the late Andy Spielman who identified the white-footed mouse as the reservoir, that is the source of this parasitic infection, and also found that the people on Nantucket had suddenly started seeing these ticks in the 1960s. Before that, they hadn't seen it before. In fact, some of the older residents called it a hurricane tick because they claimed that it came in after a hurricane in the mid-1960s. So he started working on this tick, and a prominent Nantucket resident, the late Gus Damon, he was a pathologist at Brigham Hospital, got Spielman interested in the problem and provided support, a place to stay out of his house in Sconset, and obviously served as a clinical reference interfacing with the Cottage Hospital. So Andy described the tick as a new species, Ixodes damani, after Dr. Damon, and we called it a deer tick. There are people who disagreed with this and called them one and the same, and that view has prevailed. But genetically, it's very clear that there are at least 2 different lineages of tick, and so just because I continue to use the name deer tick, and I continue to use the name Ixodes damani to make the point that those up here are very different. They actually feed on people as a subadult, as one of the immature stages, whereas the ones down south do not. And that's why you have a lot of Lyme disease up here, but very little down south. This controversy still is ongoing. So from these humble beginnings on Nantucket Island and the chain of islands, the terminal moraine islands from Long Island to Cape Cod, babesiosis has expanded, as has Lyme disease, but much more slowly than Lyme disease. But the reasons are that there are way more ticks in more places. Once upon a time, when I started as a graduate student in 1984, we said, well, you can't have this tick in stable infestations more than 30 miles inland from the coast. It requires a very moderate climate. That has changed. Obviously, the weather is changing, but also the ticks themselves are adapting to colder weather. Their natural selection is taking place, and there are lineages that are being selected for that can adapt to colder areas. They're more receptive sites. That is, if you have more ticks in more places, if you have ticks or mice transported to those sites, you can start transmission in those sites. Birds are clearly transporting ticks, and that's one of the reasons why Lyme disease expanded so quickly, is that birds themselves can infect ticks with Lyme, but it's not quite clear how babesia infected ticks can be transported. And we're still working on that. So in the 40 years that we've been working, there's still many holes in our understanding of these tick-transmitted infections. But obviously, one of the biggest factors is that physicians are better educated, people are better educated, Dr. Google is consulted frequently, and there's a better index of suspicion, there's better diagnosis and better reporting. I was fortunate to be friends with the people in the clinical lab in the cottage hospital, and they provided me with the numbers that they'd seen. The late Pat Snow there kept a record of how many cases she found each year, and the black bars are those cases that she found personally on Nantucket. And you'll notice that starting about 1992, it looked like there was a doubling of the number of cases. It turns out that's not true. The things had not changed in nature, but rather the way she did things had changed. That is, they bought an automated cell counter, and she had the counter flag any blood sample that had low white cell numbers and low platelet numbers, and spent double the time on a microscope and doubled the number of cases that she detected. So, and that's something to be aware of when you see increases in numbers of cases. Is it real? Is it due to what's happening in the environment, or is it due to people doing things more efficiently in the clinics and in the laboratories? So this is what was worked out. This is the foundation for our understanding of Lyme disease ecology and epidemiology, but it was all worked out for babesiosis. This tick has 3 free living stages in addition to the egg. What we see today, if you were to go and walk around the bushes on Nantucket, you would be picking up larval deer ticks. The larval deer ticks are the babies. They hatch from the eggs. The eggs were laid earlier this year at the beginning of the summer, and they hatched out in the middle of July, and the larvae are still around. The larvae will soon disappear. By the end of September, there'll be very few larvae out there. They are very small, about the size of a period at the end of a sentence. They are looking for things within a couple inches of the ground, such as mice, but obviously birds will be browsing or messing around the leaf litter, and other things will come along the leaf litter. But we think mice are most important in terms of feeding the larval deer ticks on Nantucket. If those larvae feed, they will drop off engorged, probably in a mouse burrow, and not come out until spring when it warms up. So the larvae turn into nymphs of next year. So what happens this year with the larvae is supposedly predictive of how many nymphs we'll have next year. We don't necessarily find that, and I'll explain why in a few minutes. The nymphs are around starting in late April. They'll be around May, June, and midway through July. And if it's a typical summer on Nantucket or other places where the deer ticks are, middle of July, it starts getting pretty dry. Dry is death on ticks, on these ticks. And so typically they disappear by the middle of July. If it's been a cool, wet summer, they can hang out until August, maybe even September. But this year it's been a very dry year, and I bet they disappeared in the middle of July. The nymphal ticks, if they fed successfully from April through July, will once they've fed, will use that blood as a resource to turn into the adult stage. The adult ticks, male and female, will start looking for animals in middle of October. It's almost like clockwork. And they're looking for larger animals. And on Nantucket, which led to the name deer tick, that animal is the deer. Virtually all adult ticks feed on deer. Adult deer ticks feed on deer. If they're lucky enough to find a host in October and November, and they'll actually be around through the winter on days when it's not cold enough so that you could be out there comfortably without gloves on. And they'll continue looking for animals until about the end of May, when they all die. They lose their energy reserves and they die. It doesn't matter whether they fed in October or they fed in May. The adult female ticks lay the eggs sometime starting in May, and all of the eggs hatch synchronously, whether they were, from ticks fed in October or ticks fed in May. They hatch synchronously in the early part of July, and then you have larvae come out again. So it's a 2-year life cycle. At the very minimum, it may be even more extended, and that's something that remains a gap in our knowledge. The way this kind of work is done is people like me as graduate students, or Joe Peisman back in the 70s, went out to Nantucket every month and trapped animals and counted the ticks on those animals. And you can see one of the graphs that was generated oh so long ago of patterns of infestation on mice. You trap the mice, you look at their ears and their nose and their eyes and under their chin, and you count the number of ticks and what kind of, whether it's a larva or a nymph. You never see adults on mice, and you'll notice that, well, the nymphs precede the larvae in the year. But that makes no sense at all, because nymphs come from larvae, and the only way that works is if the larvae of this year become the nymphs of next year. And so that's how you piece together a life cycle. So it's a lot of work, and we continue to come out to Nantucket and trap, not every month anymore, but June and September, to try to keep tabs on what the tick population is doing. Well, how is this data useful for human risk? That is, can we make a predictive model? Can we say, well, we see lots and lots of larvae on mice the previous year. That means we should have lots and lots of nymphs the next year, and therefore lots of cases of Lyme disease. And from 1991 to 1997, working with Tim Lepre at the Cottage Hospital, and 2 Nantucket college students, Heather Shampoo and Alex Weld, who worked in my lab at Harvard for those years, analyzed serum samples from fever cases on Nantucket coming into the Cottage Hospital. Normally, when a blood sample is drawn for a blood count, it's thrown away after it's used. We were able to get those after they'd been, all the identities have been marked off of them. And so we get an idea of, how much transmission there is, how much risk there is for people, how many cases there are per year. And then at the same time, we were doing our ecological studies. That is, how many ticks do we find on mice? How many ticks do we find in the bushes? And you don't really need to look at the graph in a detailed manner, but there is really no relationship even between number of cases of Lyme, number of cases of babesiosis, number of cases of HGA within a given year. And furthermore, the number of nymphs within a year doesn't seem to relate to the number of cases. And there are a number of reasons for that. One, the way we collect ticks is to go out and walk through the vegetation and drag a cloth, and the ticks think that the cloth is an animal. Well, And it's a very simple method. I've done countless hundreds of hours doing this. But the fact of the matter is they prefer animals. And so if there are a lot of animals around, you get fewer ticks on the tick collection device. And that may give you a false impression that there are few ticks in the environment, when in fact, they're actually all on animals. The other thing is, well, can we predict the number of nymphs and number of cases from the number of ticks feeding on mice the previous year? Because the infected ticks, the nymphs, come from larvae that had fed the previous year on mice or something else. And again, there was no relationship. This productivity here in the center graph is the number of mice multiplied per acre multiplied by the number of ticks that we see on the mice. And then the same year as you see that there's no relationship between what we find on mice and what we see in terms of cases. And another big part of that, of course, is, what are people doing? Human behavior is far more influential than the number of ticks in the environment. I was in Russia, in the Ural Mountains in Western Siberia with a colleague of mine who's Mr. Tick in Russia. And he'd been working on tick-borne encephalitis for his career, 40 years. He's 20, 30 years older than I am. And I asked him, Ed, you worked on TBE for a long time. Do you know how to predict the number of cases each year? And he looks at me and he smiles and he goes, very easy. So we were up in a logging camp, 6 hours drive up in the Ural Mountains in a converted barber shop in a log cabin. And I look out the window. He goes, look out the window. What's it doing? And I go, Ed, it's raining. And he goes, it's raining. Are people out in the rain? And I go, no, Ed. Are ticks actually looking for animals in the rain? No, Ed. They don't like rain. They don't like being trapped by raindrops onto leaves because it renders them susceptible to fungus. Therefore, number of cases inversely proportional to number of rainy days. And some of that is because people just don't go out in the rain. And so that has a large influence on the number of cases that you see each year. A lot of people simplistically measure this thing called infection rate or entomological inoculation rate, the number of infected ticks per minute, and try to make a story out of that. And yes, they vary from year to year in sampling sites, but you can't sample every site on Nantucket, and there are no such things as representative sites for the whole of Nantucket. But also, with the very method that you use is prone to error. That is, the same graph is there, top and bottom. The black one, though, has error bars. And the prevalence estimates actually do not differ statistically from year to year if you take that into consideration. And in fact, there's some people have asked me recently, well, we need to know what the infection rates are on Nantucket every year. And I'm going, but why? We do them. We don't do them for surveillance. And our experience in the 90s suggested it was a big waste of time to try to do this. But we do collect ticks every year. I have done so every year since 1984. I'm there in June and I'm collecting from my same study sites on the field station and on ram pasture. And we use them for other things, for other studies, for understanding deer tick virus ecology, for we've got NIH funding to look at how good is the evidence that the white-footed mouse is the main source of infection for deer ticks. And so we collect a lot of ticks and grind them up, and all of this is away in lab notebooks. It's a pain in the butt to go and get the data. But for the last 2 or 3 years, we've been fairly good at tabulating these. And you can see there's really no difference in the prevalence of infection in nymphal deer ticks from Nantucket or Martha's Vineyard. They're all roughly, 9 to 23% for Lyme. And anywhere from 5 to maybe 13, 14% for babesia. This year, though, was a big year for HGA, for human granulocytic anaplasmosis. During the 90s, we also looked at the mouse infection rates and found that I did a lot of number crunching, hoping to find a predictive model. And it turns out the number of mice infected with HGA was associated with cool, wet springs. And that's what we had this year. It was a cool, wet spring. So the ticks were more likely to be infected with HGA and statistically different from those of last year. But otherwise, the numbers that you see is proportion followed by an error estimate based upon the way we sample. And so if we go to the same site and do the same thing, drag the cloth with the same person and grind them up and do the same assays, the actual estimate could be somewhere in between those two numbers. It's a measure of sampling error, and if there's overlap, it'd be hard pressed to say that the numbers actually differ from year to year. You'll note Borrelia miyamotoi, the Asian miyamotoi disease, is very rare, as is the virus. But the virus has been around forever on these islands, and just because we didn't get any doesn't mean it's not there. We could actually go back 95%, 95 times out of 100, and we might come up with a 3% infection rate, but most of the time we'll come up with a 0. So it's there, it's just we have to work really, really hard in order to find evidence for it. And we've just decided it's just not worth doing. Nantucket has a special place in our knowledge of human granulocytic anaplasmosis. It was first called a granulocytic ehrlichiosis. It isn't ehrlichiosis. Anaplasma is an infection of bovines and infects the red cells. Ehrlichiosis or granulocytic anaplasmosis infects leukocytes or white cells, has no red blood cell component, whatever, and there's a little controversy over the names. And interestingly enough, I'm one of the controversial people saying I don't use the term anaplasmosis. But anyway, with Tim Leprey's help and Pat Snow's help, we were able to find the very first case of HGA in the Northeast. Pat Snow, I gave her the word, anyone who comes in with this set of signs and symptoms, look at the white blood cells, and lo and behold, she sends me a blood sample, and we were able to get it going in the laboratory, in laboratory rodents, using methods that came out of a paper written in 1938 by Ernest Edward Tisser, who was professor of comparative pathology at Harvard Medical School, one of the first chairs of my old department there. He was looking at infection of voles on Martha's Vineyard, came up with this new infection in white cells, but then parenthetically in the text he says, oh, and I see babesia too. And so babesia is not young, it's not recently introduced. It was at least, we know it was there in Martha's Vineyard voles in 1938, 1937. And in fact, that paper stimulated me as a doctoral student to go to museums and use a new technology at the time, I'm dating myself, called polymerase chain reaction to find DNA of Lyme disease bacteria in ticks from 1942 from the end of Long Island and mice from Cape Cod from 1896. And so these infections have been around for a long time, probably were here before the Ice Age, and were pushed down into little sites at the edge of the ice sheets and then came back as the ice sheets retreated. Well, I said I was in Russia. The reason I was in Russia is because the tick there is very, very similar to the one we have here. It was known for transmitting tick-borne encephalitis virus. Tick-borne encephalitis virus is a relative of West Nile, dengue, yellow fever, Zika virus. And there, it was a significant infection in terms of public health. As many as 40% of the people who got it died. It is one of these biosafety level 4 organisms where you need a spacesuit facility to work with it. But they also had in the same sites with TBE virus, HGA, Babesia microti, and Lyme disease. And my thought was, well, they come in systems like this. They probably predated the glaciers. If they have a tick-borne encephalitis group virus in Russia, where we see Lyme, Babesia, and HGA, we should also see a tick-borne encephalitis virus, and very easily found a tick-borne encephalitis virus in 1996, and I called it deer tick virus. Sadly for me, I had people like Tim Lepre beat the bushes on Nantucket because we knew it was there, and we found it, you know, the very next year, 1997, on Nantucket. Once we knew what we were looking for, and I told Tim, well, look for encephalitis, look for meningoencephalitis, neck stiffness, paralysis, and he's never found anything. I gave up. I had other collaborating physicians in New England working with me too, and just decided, well, it's just a variant that doesn't cause human disease. Oh well. And then I moved on and didn't do anything until this report in 2009, which found genetic information from deer tick virus in the brain of a fatal case from New York. Everybody calls it Powassan virus. It is related to Powassan virus, but like the deer ticks themselves, there are two lineages that are quite distinct genetically, and they have different ecologies. So it's around. We find it, again, on Nantucket, we can find it in anything from 0 to 3% of the ticks, depending on how many ticks we look at. And so it's around. But one of the things we're trying to understand is, you know, why is it that we don't see cases in places like Nantucket and even Martha's Vineyard? The Vineyards actually had 2 cases, but they have this woodchuck tick, which is the vector or the transmitter for real Powassan virus. We know deer tick virus is present, but not Powassan virus on Nantucket. And we have an NIH-funded program trying to understand whether there are lineages of virus that are less likely to cause encephalitis or fever in people than others. And again, this is one of those very difficult viruses to work with. I have a biosafety level 3 laboratory and have to do all of my work there. But you'll note, you know, in the late 90s, we were getting infection rates from Nantucket, for example, that are very, very similar to what we estimate today, anywhere from 0 to 4%. So things are very, very stable on Nantucket. So that's a little bit of the history of the knowledge that we accumulated as a result of our visits to Nantucket and working at the field station and on Ram Pasture and working with Tim Lepre at the Cottage Hospital. And accumulation of knowledge is nice, but you know, as a public health person, I want to see what we can do to reduce people's chances of getting infected by one of these pathogens. For many reasons, one of which being that Jim Lentowski has been in the bushes a lot and has suffered from some of these infections, but also because we tapped him every year. We would hold this Harvard medical entomology trip at the field station where we bring students and tell them about the history of babesiosis and Lyme disease and the ecology out there. And Jim would always speak to us about land use out there and how it influenced the ecology that we see today. Well, he was one of the first in the country to actually educate visitors about risks. And this is critically important in terms of trying to reduce risk. That is, if people are informed, they are empowered. And he'd have these beautiful signs. This is more recent. In the old days, it was not as fancy. But if you just remind people that there are ticks around, that they should wear repellent, and that they need to call their healthcare provider if they feel sick after being on Nantucket or while being on Nantucket, that goes a long way to reducing any long-term complications from having pretty much infections that are fairly easily treated if you catch them early in the first place. But what we understood from the ecology of Lyme disease on Nantucket is that there are two facets that we need to keep in mind when we're trying to attack the problem. One is, and so keeping Lyme disease bacteria around from year to year in nature requires two different things, and they're largely independent. That is, you have production of ticks, which is, tick reproduction. The female tick feeds, the blood becomes eggs, the eggs hatch and become larvae. And interestingly, the larvae do not inherit infection from the female tick. And the source of this blood meal, and only female ticks lay eggs. The larvae do not, the nymphs do not, the males do not. It's strictly the female tick. And the source of that blood meal for the female tick is a larger animal. They will not feed on mice. They will not feed on any bird. It has to be something a cat size or bigger. And in fact, they don't even like cats that much. And usually it's deer, dogs, coyote, bear, moose, obviously humans. But of all of those larger animals, it's only deer that are most common on Nantucket. Yeah, there's some feral cats around. There used to be feeding of ticks on dogs, but within the last 15, 20 years or so, there have been a number of products out there that allow people to either prevent tick bites or kill the ticks that are feeding on the dogs. Hence why we don't see dog ticks, the famous big beach grass tick, which used to be at the end of Nantucket Road, was a great place to look for dog ticks. Dog ticks have literally disappeared from Nantucket because most people are treating their dogs with these anti-tick preventive things. So adult ticks do not feed on rabbits, they do not feed on mice, they don't feed on squirrels, don't feed on birds. But the second aspect of this is because the tick is not born infected, it has to become infected. And it does so by feeding on an animal that is infected. White-footed mice, shrews, rabbits, squirrels, and some birds are known to infect ticks. They also feed on a number of other animals that do not infect them, which is why, for Lyme, the infection rates, the proportion of ticks out there that are infected by the bacteria that cause Lyme disease is roughly about 20%, which means 80% of the blood meals must come from animals that are not infectious. So those are two different processes, and you need to have specific means of attacking each of those. There is no one method to do both at the same time. The picture on the bottom right is of an egg mass from a fed female deer tick. That's 2,000 eggs from a single female deer tick. You can do the math. If you can have, I've seen as many as 100, 200 fed females come off of a single deer. Let's pretend that it's maybe 10 fed females per deer per week. That takes a tick a week to feed completely and drop off. And the ticks are around for about 16 weeks. So it's 16 times 10 times 2,000 eggs from a single deer, and then multiply that by how many deer you have. Therefore, it's no surprise to us when we see analyses like this that look at the spread of Lyme disease and how quickly it spreads across eastern Massachusetts during the early and middle 2000s. The sites in red are those towns that report more than 5 cases. And then down below, the two maps that you see there are taken from an interesting article from Massachusetts Division of Fish and Wildlife, which looked at where people can effectively hunt and thereby manage their deer herds. There are towns that actually have no hunting whatever. And others have special restrictions. And the most important part of this is that there is statute in Massachusetts that prohibits the discharge of a firearm within 500 feet of an inhabited home. without written permission of the homeowner and all abutters. And so each home takes away 18 acres of land that can be hunted. And that's why most of eastern Massachusetts is orange. That's where this firearms discharge setback is, which means you cannot manage deer well within 495. And we've seen this dramatic increase in the number of cases, and I don't think it's a coincidence. And the reason I don't think it's a coincidence is because as part of my doctoral work, we ran the first study of deer reduction. Lewis Bay, Great Island, is the peninsula on your left as you're coming on the steamship out of Hyannis. It's got a little lighthouse on it, Point Gammon. And the people there were people of means. By 1980, 16% of them had Lyme disease, and they said, enough's enough, we want something done about this. They came to my boss, Andy Spillman, and said, we want you to do something. And he said, okay, we'll kill the deer and see what happens. At the time, Alan Steer, who discovered Lyme disease in the US, was working with us as the clinician, and he was able to determine that there was a bona fide case of Lyme disease as early as 1962 on Great Island. And we tracked cases every year by a twice-a-year blood draw. When I took over the project in 1984, they would have their little church service out on in the middle of the woods there on Sundays, and the hour before their church service started, I had a table set up, and they'd put their arm out, and I'd bleed them. And I'd bring the blood back to the lab and analyze them for antibody to Lyme and babesia and everything else. But at the same time, I was also trapping every month from April through October, same as Joe Peisman did and how we continue to do in some sites, to try to figure out how many ticks were on mice. And you can see that in the upper right graph, Great Island, you see a precipitous decline in red bars. Most of the deer were removed starting in 1982, 1983. And by 1984, there were no more than 6 to 10 deer on a peninsula of roughly a square mile. They can come on and off as they please. But the caretaker has been instructed to keep the herd down to fewer than 8 animals at any one time. Whereas Nantucket, the field station, and Ram Pasture were our control sites because there was no active effort to really reduce the deer herd. And you can see there that the tick numbers are pretty stable on Nantucket over the period of this study. We noted that for babesia, we could probably make a case for a reduced prevalence of infection in mice, and also the bottom left corner graphs show that there were fewer mice that were positive for Lyme antibody after the deer were reduced on Great Island. Compared to Nantucket, where there was no change whatever. So we think this is as good as the evidence gets in terms of a relationship between deer ticks and transmission of these infections in nature. Now, the human cases, you'll see that there are very few human cases after 1984, 1985. We'd like to attribute that solely to deer reduction, but note that I was out there every month from April through October. I would talk to people, they'd come by and get bled. I helped educate them, and that certainly helped with reducing risk. So to be completely honest, it wasn't just killing the deer. And note that it wasn't killing all the deer. There were still deer on this peninsula. It's a matter of reducing them by 80% or so. And this is really the objective of deer reduction. It's not to eradicate deer, although Nantucket could eradicate deer, and that should pretty much take care of the problem. But the idea is to return the landscape to what it used to be when you had little patches. Now, the picture there, on the bottom right, was taken in the 1970s from around Altar Rock. You'll notice now there's very little grass and pasture up that way. The vegetation has changed and allowed deer and mice to spread from these little patches into bigger and bigger patches. And the idea of deer reduction is to push them back into little patches where people are much less likely to step in into the mess of ticks that the deer are producing. So it's not a matter of eradicating deer, but rather pushing them back into smaller and smaller areas and having fewer of them drop ticks and contaminate more of the environment. People say, well, but if you kill a deer, we got so many rabbits, and rabbits are loaded with ticks. And yes, they truly are loaded with ticks. I counted, I looked at enough rabbits on Nantucket and looked at 3,600 ticks and found that there were very few deer ticks on them. They were all a relative of the deer tick, Ixodes dentatus, a rabbit tick. And in fact, there's a T-shirt, I believe, from the Mariah Mitchell that has a picture of a tick. It's actually a picture of Haemaphysalis leporus palustris, the other rabbit tick, not of Ixodes damani, the deer tick. But anyway, there were two major publications on Nantucket rabbits, one done by me and one done by my wife during her doctoral work on the field station. And we know that adult ticks are not found on rabbits. So if you remove deer or reduce deer, they're not going to leap onto a different host. And we found the same thing on Great Island. That is, we did nothing to the foxes and skunks and raccoons and possums that were there on Great Island. They continued to persist, yet we maintained the tick population at 20% or less than what it used to be before deer reduction. I go back every spring and every fall and have done so, since 1985. I was just there in April, and I will be going again in about 6 weeks and sampling adult ticks, and the numbers remain very low. In fact, last year I could hardly get any. But they haven't disappeared. All you need is one female tick to reproduce, and you can keep the tick population going. So if anyone tells you they can eradicate the tick, you can just laugh at them. It doesn't take much to keep the population going. What we want to do is cut down on the number so that people are less likely to see them on a regular basis. So so I said the Nantucket was instrumental in rolling out the first human Lyme disease vaccine. How did this come to be? Well, I was actually a postdoc at Harvard at the time, and another postdoc down at Yale had done an experiment where he developed a simple vaccine against Lyme, and had actually gotten a paper in Science about it, and we just sort of laughed at him because we said, oh well, the Lyme disease is transmitted by a tick. It's not by a hypodermic syringe. If you actually used a tick, your vaccine would fail. And to his credit, he invited me down, said, bring your infected ticks, we'll test our vaccinated mice. And lo and behold, it worked, but it worked in a way that neither of us, neither Errol or Fickrig or I anticipated. That what it did was to kill the Lyme disease bacteria in the tick. So a vaccinated animal has antibodies against the major outer surface protein of the Lyme disease bacterium. And that antibody is circulating in the blood of a mouse or a human. And as the tick feeds, the antibodies coming into the tick as it's feeding with the blood meal and bombing the bacteria before they have a chance to get out. Not only that, you can render mice non-infectious by giving them antibody, and then once that antibody disappears from the blood of the mice, they become infectious again. So this was the basis for the human Lyme disease vaccine Lymerix, and it was the basis for baited vaccines to be fed to mice, and it's also the very mechanism that we hope to exploit with a genetically modified mouse, which I'll talk about in a minute. But the whole point about the Nantucket being the site was that SmithKline knew we were working on the islands, and they came to us and said, we want to try a Lyme disease vaccine. And my job as a postdoc was to go out to places like the Vineyard and Nantucket and Block Island and try to drum up volunteers. And I made this poster, and I put it in the liquor stores on Nantucket, thinking that was the best place for people to see it. As it was, we had a hard time getting people to volunteer, but nonetheless, Tim Lepre was able to get 100 people or so from Nantucket. We got 100 from the Vineyard and 100 from Block Island, and found that this vaccine was well tolerated. Yes, they had side effects, but again, those people who claim that Lyme disease vaccine is bad for you need to look at the side effects and complications for any other vaccine, and there is no difference whether you're looking at Lyme disease vaccine or papillomavirus or anything else. And at the time, we also got very good data on how effective it was. That is, it was 50 to 80% effective, and that led to a large trial run by Alan Steer with over 8,000 subjects. And the vaccine was put on the market for a couple of years. The market was not as good in the late 1990s, the early 2000s. Lyme disease was still fairly restricted in its distribution. There were fewer cases, and the CDC wasn't terribly wild about the vaccine, saying, well, you know, Lyme disease is easily treatable. It's not really high up on our list of things to push for. And so the company didn't make any money from this vaccine, and they were hit by lawsuits from people who claimed harm from the vaccine. And they just threw up their hands and said, we're not making money. We spent a lot of money on this. We're just getting out of the business. And the sad part of it is it sits on a shelf. It actually sits in a freezer in Belgium, where SmithKline's, now Glaxo's research headquarters are. And it's the only intervention ever proven to reduce the risk of Lyme disease. It clearly showed that you could cut down the number of cases. And some people will say, well, but what if it's only 50% effective? There's a difference between a vaccine that's 50% effective at the individual level, where you can say reasonably, well, it's sort of like flipping a coin. If I take the vaccine, I still have sort of a chance of getting infected. But for a public health person to look at a community and say, wow, we dropped the number of cases by 50%, that is a fantastic public health achievement. So I am disappointed and angry that something that I contributed to as a young man, two things that I contributed to as a young man, deer reduction and limericks, could have prevented hundreds of thousands of cases of Lyme disease across the Northeast. And one of them we can't do because people like Bambi, and the other one we can't do because the lawyers killed the company's economic incentive. Well, I still think that we can repurpose the old vaccine. It was proven to reduce risk, and I've been trying to find investors for reviving it under a nonprofit model, but the oomph in that has gone away because Valneva, a French company, is in advanced phase 2 trials with a very similar vaccine. I think it should work. They're going to be spending $150 million to bring this thing to market. Their timetable is a rollout in 2024 if all things go well. I don't see why it wouldn't go well, but we just don't know, and it's nice to have something else in the pipeline. But anyway, I've sort of let this take a back seat now. 3 years ago, I was very interested in setting up a nonprofit to produce the old vaccine and deploy it. I'm even tempted to put the recipe out on YouTube and let people make it. There are a number of do-it-yourself biologists who work in their basement. Anyone with a rudimentary knowledge of recombinant DNA technology can make this vaccine. There are people who are fed up with the price of recombinant insulin for diabetes and are making their own insulin in basements. And so this is an interesting new model for circumventing some of the costs of healthcare. So we've heard some press releases about baited vaccines. You feed mice the essentially the Lyme disease vaccine, they get antibody and they're no longer infectious to ticks. We were working on such a project ourselves with a live vaccinia virus expressing this protein, similar to what we see for rabies control in raccoons. There's another company who is way far ahead. They've actually got a commercial product. We don't think much of their product, but you know, every little bit counts. But there are other ways of approaching this, and the evidence for white-footed mice being important come from a number of papers, not just those from the field station, but also up at Crane Beach, but all out of the Harvard lab. And they're based upon the fact that mice are the most common small mammal. They have lots and lots of ticks on them relative to everything else. And maybe 96% of all the larval ticks feed on white-footed mice. So they're a logical target for trying to reduce the number, you know, trying to reduce infection. So it has nothing to do with producing ticks, but rather once ticks are produced, try to prevent them from getting infected. So Kevin Esveld, a very, very smart young faculty member at MIT, reached out to me and said, hey, you've been working on mice for a long time on places like Nantucket. I want to try this idea that I have in making mice so that they are constantly expressing the antibody that was the subject of Lyme disease vaccination, OspA antibody. And I said, sure, we need to try whatever we can to provide as many tools as we can to reduce risk for people. Putting our eggs in one basket, saying, oh, we'll wait for the vaccine and that'll take care of the problem. We then don't have to go down the unpleasant road of trying to kill deer. We don't have to do anything else. We don't even have to take precautions anymore. We'll be vaccinated. That is very bad public health. We need to have a quiver of arrows that communities can select from and use and tailor to their own purposes. And so I find this idea very interesting and very forward-looking, because Lyme disease, we know the mechanism of action for vaccination. We know it's effective. It works in animals and it works in humans. So it's a good model to start with this technology. But my interest as a tropical public health person is to deploy it against things like Lassa fever or rhinovirus infections in the tropics. So this is a proof of concept experiment as far as I'm concerned, but it can also provide a great tool for communities who don't have the guts to deal with their deer problem, who can't educate their community in methods of preventing tick bites, and there are plenty of them. I mean, I've crawled around the bushes for over 30 years on Nantucket. I've never had Lyme disease, even though I'm picking ticks off of me left and right, simply because I'm vigilant. I check myself very carefully. I take a shower the moment I'm out of the woods, and I watch. And I also have an advantage in that I itch when I get bitten by a tick, and I know they're there, and I pull them right out. Nonetheless, almost every year I do take prophylactic antibiotics, and that's a very simple thing to do. There was a placebo-controlled study published in the New England Journal of Medicine. After any recognized tick bite in an endemic area, 1 tablet of doxycycline is sufficient to reduce your risk of getting Lyme disease by over 80%. And so I have most years had to resort to the single tablet of doxycycline because I let a tick get through and actually feed on it. Anyway, the idea is to make mice and test them in the laboratory to see that they actually fail to infect ticks or fail to become infected, and then do a field trial on a small island. The regulatory agencies, and this is probably the hardest part of the project. The MIT people are very close to making the mouse. They've actually made a laboratory mouse doing this, and now the challenge is to do it for white-footed mice, which are more closely related to hamsters than they are to lab mice. But the regulatory agencies, EPA, FDA, CDC, U.S. Fish and Wildlife, Mass Wildlife, all of them will have to review this project, and the amount of red tape that's going to be required will probably take us longer to solve than actually making a mouse. But nonetheless, we're hoping to do a field trial in maybe a year or so, a small island somewhere, to have a study of its efficacy, how many mice become infected versus in a site where these mice have been introduced versus a site nearby where the mice have not been introduced. And if that's reviewed and permits are provided by federal and state agencies, then we would come to a place like Nantucket and ask the community if this is something that they want to consider for their quiver of arrows. So it's not a done deal, but nonetheless, we continue to do this because it has broad range implications for public health elsewhere. Anything having to do with a rodent transmitted infection could be addressed with this. And that's why we're so happy that people on Nantucket and Martha's Vineyard have been so supportive of this project, even though there's no guarantee that it'll ever come to either of the islands. The communities have to vote. Kevin has been very, very good about being transparent about this and involving the communities in the decision, that we would just walk away if the town said, no, sorry, we're not interested. But that doesn't mean we shouldn't do this because it may have future application. So obviously all the work that I put in during from 1985 to about the mid 2000s, when I had a lot more time to come out to Nantucket to the field station, stay at the field station and do my trapping for 2 nights a month, every month from April through October. We accumulated a lot of data on how many ticks are on mice and what the patterns are. There is no pattern of increase. The tick population is stable out there. And we're doing the same sort of thing. We're looking at mouse demography on a number of candidate sites. Tuckernuck is listed as a field trial site, but we respect the Board of Health's suggestion that Tuckernuck is part of Nantucket and therefore should not be used as a field site. So Tuckernuck will not be used as a release site initially, may not ever be the site of a release, but there are other places that we're working where we could actually do a preliminary experiment. So the timeline probably has changed. We have not been able to make as much progress as we want in the laboratory. Again, this is not cheap work. We've been very fortunate to have the Rainwater Foundation interested in this, and their support has been critical in getting us to the milestones that we have today, but like Manhattan projects everywhere, the more funding that is available, the more hands that can be put to use. You'll notice I'm grooming my own hands. My daughters help me trap, and in fact, they're learning all about tick collection and how I go about processing animals. But I want to wind up by acknowledging the National Institutes of Health, who have generously supported our field studies through traditional investigator-initiated grants. The Egan Foundation on Nantucket through Tim Lepre and the Gordon and Laura Gunn Foundation provided small grants during a time that was very lean for a lot of scientists during the recession. America had to tighten their belts, and scientists did too, and NIH was very difficult to get money from. Fortunately, we're back in the saddle. We have NIH money again, and like I said, the Rainwater Foundation has helped us tremendously in trying to accumulate the baseline information that we need to go to the regulators and say, let us do a small pilot study. But none of this would have happened without the many Nantucketers who have helped us through the years. The late Wes Tiffany, his wife Susan, Tony and Terry Molis, Jim Lentowski, and of course Tim Lepre is Mr. Tick, and Mr. I believe he's still the coroner out there. He's everything on Nantucket. The Conservation Foundation has been very gracious about allowing access to all of their properties from the very start. But none of this would have been possible without the field station. It is impossible for us to stay on Nantucket during the summer if we had to pay the same rates that tourists do. Yes, we may have funding, but when you consider that it would cost us, maybe $1,000 to do a monthly trapping, that $1,000 could go towards paying an undergraduate or a part-time technician in the laboratory for 2 weeks or more. And so it's a bad way to spend money on research grants, and we've made tough decisions over the years, saying, well, sorry, we just can't go to Nantucket as much anymore, because it just if we didn't have the field station available. Also, the steamship has raised prices, and getting a field vehicle over is tough. But nonetheless, we've made things work. We continue to work out on Nantucket, and even though you may not know who I am, I'm there. I'm in the bushes. I deliberately don't attract attention because we only have 3 days to work, and I need to make every hour count when I'm getting data. So I'll stop there. I've gone a little over time, but I'm hoping that there's some feedback or some questions about the things we've been doing and the things we hope to do.
Well, thank you so much. Can you unshare your screen?
Yeah.
Thank you. Excellent. Okay. Thanks again, Sam. That was a lot of information and really interesting. In particular, during this time that we're in, there's so many people that are, wanting to be outside, going outside, and rightly so. We all want to enjoy the outdoors and be healthy and happy and socially distant. But I am concerned, and this is why we really thought this would be, as usual, a great talk, because Lyme and ticks and all of the other tick-borne diseases are an issue for us here on Nantucket and in New England. But in particular right now, where we really have a lot more people going outdoors to spend their time, and perhaps more people than normally would be outside, probably need a little bit more education if they don't normally spend this much time outdoors. So I thank you very much. This is an important topic, and something that we'll revisit. We have a few questions, so I would like to pose them, but also know that we don't have endless time. So I will start with this one from Dr. Malcolm McNabb. He's referring to the Nantucket Infectious Disease Report from 2009 to 2018. And he asks you, when you look at that and you look at the graphs, there's a definite decline of Lyme disease in the last, since 2013. It looks like it spikes and then it tapers off after that. With that in mind, can you comment on what you think about that? I mean, I guess
well, getting that kind of data is tough. Sadly, the Department of Public Health has gone a different way in terms of their surveillance and aren't accumulating the case reports like they used to. But let's assume that the data is correct and the numbers have stabilized or in fact are going down. I have to look at that as a wonderful sign of people educating themselves. I see no evidence that the risk in the bushes has changed over 30 years. There are plenty of ticks. If anything, there are more ticks in more places, and there should be more cases. As in this, one of the sad things of working on Nantucket for so long is once upon a time you could park down at the A&P and leave your car there for weeks. But now, of course, you get ticketed and towed. But all the houses that have gone in, the development that has happened on Nantucket means there are more people's homes right up in the bushes where they should be exposed to more and more ticks. So something has balanced that out. And I think it's because of efforts of people like Malcolm and Roberto Santa Maria, the Board of Health, just the general fact that there's more information out there on how to prevent tick bites. I'd be awfully surprised if someone coming off the boat has never heard of ticks. There are good products out there, repellents, plain old DEET works just fine. But now you can buy clothing that has already been treated with permethrin, which is death on ticks. Our military uniforms have been treated for 20, 25 years, and our soldiers spend 24/7 in those clothes. I am so fearful of its effects that I can't have it anywhere near my car because I want my ticks alive. But you can buy this stuff already treated from Insect Shield, from Cabela's, from Beans, from Eddie Bauer, so people could be using those. I know people are pushing out the message of checking themselves for ticks in the shower. And I'm hoping that everybody's getting checked out for fevers. And so I think the educational efforts and some of the very basic tools that we have in repellents and clothing treatments are making a big difference. Over on the vineyard, we see a reduction in number of cases as well. Some of that is due to this policy that we've promulgated over there, which is the 1 tablet doxycycline response to any recognized tick bite. There are clinics there. You walk in, you say, I got a tick bite. They just hand you the doxycycline and you go on your way. And that clearly reduces risk for Lyme and maybe for HGA and Borrelia miyamoto. Sadly, it doesn't do anything for babesiosis, but all of these factors have balanced, I think, what should be, if you take the ecological signs into consideration. The ecological signs suggested that we actually should see more cases because, again, there are more houses, more people in the bushes, more habitat, yet we don't see a rise in number of cases. It's remained stable. Now, does that mean people can let their guard down? No, of course not. We could have a bad year. There may be a perfect storm set of conditions that may produce a bumper crop of infected ticks, but I find that unlikely because I've been working on this for a long time and I've never seen that. People wave their hands about global warming, but if anything, global warming is going to make fewer infected ticks than more infected ticks. The Lyme disease bacterium doesn't like warm weather. The tick dies from being dry and hot. So I still think that there will always be a problem. The question people really should have is, well, how much of a problem do we want to tolerate? And the biggest issue of that is children. If you could reduce the effort that it takes every single day as a parent to worry about your child, to check them for ticks, to remind them to use repellent, to tell them not to go play in the bushes, if you could find some way to reduce that by half, then there should still be a lot of interest in doing other things besides waiting for a Lyme disease vaccine or hoping that a downward trend continues and everybody becomes educated. Not only that, we found 5 different infections in deer ticks. For all I know, there could be others lurking. I'm on my nose is to the ground, and I have hints for number 6 and number 7, but that's for there's no evidence that I can share yet with that. But so it's not that Lyme disease is the end all and be all. It started with babesiosis. There's relatively little you can do against babesiosis. There is no prophylactic treatment. So you still need to come to grips with the other infections. And so broad application interventions such as deer reduction or modifying animal reservoirs so that they're no longer capable of infecting ticks with these other agents. Those are things for the future. One of the reasons I stress things like deer reduction and perhaps modification of mice is that they're more permanent solutions. Once you reduce the deer, it's a maintenance phase. You don't spend as much energy and resource keeping the deer numbers down. Once you do a population replacement, hopefully there are ways of keeping that so that you don't have to expend energy or resources continuing to replace. But things such as vaccinations, tick repellent checks, you do have to do those every day. They're what we call short-term methods. And once you stop doing them, the protective efficacy goes away. And so we need a combination of short-term methods and long-term methods to look for the future, to try to make things better for our grandchildren. And I think that's really the target, is what do we do as a community to make things better for our grandchildren?
So you think that even if the numbers continue to decline and people are educating themselves and adhering to these practices, we should still have thoughts about using a genetically modified mouse?
The community should still have thoughts about doing as many things as they can to complement education and awareness. This is not going to go away. It will always be there. And again, I don't know that there's anyone who would stick their neck out and say, well, okay, 100 cases a year is a perfectly acceptable number, or 50 cases a year is a perfectly acceptable number.
How many deer do we have on Nantucket? Do you know? And what would we have to do to significantly reduce the population? Or maybe this is the topic for our follow-up.
Yeah, that would be a good one for the follow-up, but let me just say that empirically, we know that deer reduction has not worked on Nantucket. We tried very hard to do this back in the mid 2000s. A lot of effort was expended on trying to bring this to Nantucket. The politics are such that it would never be supported. However, I am working on ways of circumventing that by trying to find a way to change legislation so that people can make some money from deer. That is, to be able to sell venison. Can you imagine what Nantucket venison would go for in a restaurant? So I've identified the legislation that needs to be modified. Apparently it'll cost $100,000 to get a lobbyist to insert the language in the right place. But to get exemption from the one problem there is in selling venison, which is the mode of killing must be inspectable. That ruins the idea of hunter-killed venison. But imagine if families could take advantage of a commercial season on deer and supplement their income with a very healthy meat. So, I continue to look at ways where it's a win-win for everybody.
Well, I thank you so much. We're very lucky to have you devote your time to educating us with so much information. It's really very interesting. And as I said earlier, we'd like to have a follow-up discussion about some of these things you've brought up where we can get into it in a little more depth. So for those of you that are tuning in, either still live or watching this in a recorded fashion down the road, please do type in your questions or make some comments that we can collect up and look at. But I am very grateful, and thanks again, Sam. This was really great.
No, thank you, and thank the UMass Field Station, because it never would have happened without your support.
Well, you're very welcome.
And see you soon, I hope.